Hi learners, welcome back. I am Dr. Gurunathan from Krista Jayanti College and I welcome you for today's session which is about biological databases and introduction. What are biological databases? Basically, it is a collection of biological data and the data that is collected is organized in a manner which can easily be accessed and it can be managed and it can be updated on a regular basis. So what kind of biological data are we talking here about? We are talking about two important macromolecules. One is the nucleic acids, which encompasses your DNA and your RNA molecules, and the other important macromolecule is your proteins. And we are focusing on two important aspects of these molecules. One is either the sequence information or the structural data that is associated with these two molecules. And as you can see here, basically a biological database is in the form of a table where you have rows and columns. These rows are also referred as records and the columns are also referred as fields. And if you take a single row, that would represent multiple aspects associated with one particular, say, information. And whereas when you speak about the columns, which are the fields, they are individual entities that are associated with each of the data that is existing there. Next important aspect, what kind of DNA sequence data you require. See, as you can see here, the DNA has four important molecules. One is called as adenine, which is represented as A. The other one is called as thymine, which is represented as T. And you have guanine, which is represented as G. And you also have cytosine, which is represented as C. So arrangement of these is actually called as a sequence, and your biological databases can store these information. And whatever we are speaking here about the purines and the pyrimidines of the DNA molecule or even the RNA molecule, for example, in case of RNA molecule, you will have uracil instead of thymine, and to represent uracil, we use the alphabet U. So all these are called as single letter representation. And the arrangement of each of these nucleotide molecules in the DNA is called as DNA sequence data. Whereas when you move down to the polypeptide sequence, you have different amino acids. Particularly, you have 20 standard amino acids which are forming the entire protein molecule that are functioning in cells. And these individual amino acids which are present in the proteins are also given with alphabetical representations, particularly single letter alphabetical representations. And each of these amino acids can be understood. And if you are going to have a sequence of these amino acid alphabetical letters, we call it as polypeptide sequence. The next important detail, that is about the structural detail. See, we always see that the biological macromolecules, they are having specialized structures, and these structures are usually three-dimensional in nature, and these three-dimensional structures should be available for them to be functional. If they are not having the three-dimensional structure, if they don't form the three-dimensional structure, they are generally considered to be non-functional, and because of that, your biological databases are also storing structural information. And you can see that in 1965, a scientist called Margaret Dayhoff developed the first protein sequence database, and it was named as Atlas of Protein Sequence and Structure. To the classification of the databases, we are speaking about classifying the databases based on three important characteristics. First one is based on its content. And based on the content, biological databases are classified as sequence database, or they can be classified as structured database, or they can be given as other databases. And as you see here, the sequence database can also be further classified into nucleic acid databases and protein databases. Whereas when you speak about the structured databases, you don't have that classification, but you have some examples such as PDB. You have a structural database called SCOP. You have structural database called as CATH. Similarly, you also have in other databases where the contents that are existing in it focuses on other details. For example, if you take about REBase, this is actually a database which is storing information about the restriction enzymes that are known to us. Whereas when you speak about GPCR database, they focus on G protein coupled receptor database. So you see that this classification is a widely accepted one, and this classification is also a very simple one for many of us to understand how the biological databases are classified. Moving on to the classification scheme, which is based on the biological data source. That is a second method of classification. We have three different databases available. One is called as the primary database, which is focusing on the data that is obtained from experiments that are performed in the laboratories. Similarly, you have the secondary databases, which are also called as derived databases, because these databases, they derive the information from the primary database, and they are giving us more information pertaining to the data that is stored in them. For example, if you take a database called SCOP, which is the acronym for Structural Classification of Proteins database, they derive information from the primary database such as SwissProt and PDB, and this contains additional information which is not available in your primary database. You also have the third group of database, which is called as composite databases, and this database amalgamates a variety of primary database sources. For example, a well-known composite database is NCBI. NCBI stands for National Center for Biotechnology Information, and this encompasses several different databases. For example, you have something called as PubMed in it, which contains exclusive literature information. Similarly, it also has a database called GenBank, which stores exclusive sequence information of nucleic acids. Similarly, you have a protein database in it. Similarly, you have several other specific biological data containing databases. To the next important information about the three primary databases that are known to us is NCBI, the other one is DDBJ, and the third one is European Nucleotide Archive. All these three databases, they are now into a common collaboration, which is called as INSDC, International Nucleotide Sequence Database Collaboration. The main purpose of this collaboration is anybody who is going to provide the data into one of these databases will be shared with the other databases by the end of the day. So this has reduced the requirement for uploading the data by the author to all these databases. Instead, if I upload in one database, that will be automatically shared, and this reduces the redundancy that could occur when the authors are directly submitting data to the databases. Down to the next slide is just showing you the screenshot of another important primary database, which is called as XPASI, and this database is actually maintained by Swiss Institute of Bioinformatics, and it contains protein information and also tools that are required for handling the protein data. Similarly, this is another important primary database which stores structural information. It is called as Protein Data Bank, also known as an important information portal to biological macromolecular structures. And this database is slightly different from the previous one that we have discussed, that is the XPASI, because this database stores protein structural information, whereas the previous one stores mainly sequence database or sequence information, and this is accessible in the website called www.rcsb.org. Moving down to the next group, which is called as UniProt, which is a combination. UniProt stands for Universal Protein Resource, and it is a combination of three other important databases existing. One is called as SwissProt, the other one is Protein Information Resource, the third one is Translated EMBL. The screenshots in the secondary databases, you have a typical example of a ProCite, which is also available as a part of XPASI, and this is a database which contains information about protein domains, families, and functional sites. And this screenshot is a typical example of a composite database that I have already mentioned, that is NCBI, and it is an interesting database for us to also